Okay, boys and girls, we are back. Hello. Now, we're going to be starting chapter 5 here, where we're going to be examining the plasma membrane, what it takes to put together the membrane, and what's involved in maintaining said membrane. Now, you got to remember, plasma membrane basically has four functions. Four functions are: one, to act as the boundary. Here is the boundary. Here is the fence. Here is, the dividing line. Inside, outside, outside, inside. Not only is it acting as the boundary, it's acting as the gatekeeper. What can get in, what can get out of the cell. We're going to see that certain things can move through, other things have to be moved through. Also, the plasma membrane, it's going to have certain proteins, glycoproteins, carbohydrates associated with it that are going to take signals from outside the cell and bring them in. Because remember, the cells have to be able to talk to each other. They have to be able to communicate. They have to be able to respond to what's going on in their environment. And then there's adherence. Two cells have to stay stuck together. If they can't stay stuck together, you don't have a tissue. No tissue, no organs. No organs, no organ systems. We have what we refer to as a fluid mosaic model. Fluid, not water fluid, but fluid as in always in motion. Okay? Things are moving, things are twisting, things are pointing out, pointing in, pointing out, pointing in, back and forth and moving around. Fluid. The mosaic comes from actually the art form known as mosaics. Now, you've seen tile mosaics, pottery mosaics. They take and they crush up into small little colored pieces, either tiles or plates or glass or something, and then they stick them together so that when you look at it from a distance, you see a picture. When you get up close, all you see is individual little shards. Well, that's what's going on here. Turns out your plasma membranes are not just ubiquitous phospholipids. There are different types of phospholipids that come in different flavors, as we're going to see in this chapter. And also interspersed all throughout are going to be, as we see here, proteins. Proteins and different types of steroids and cholesterols, and then different sugar things. All of these combined with the phospholipids gives us our mosaic, our bigger picture. Now when it comes to the phospholipids, as we talked about in a previous chapter, there's going to be three main parts. Sorry, I showed four. Three. The phosphate group at the top, just below it the glycerol, and then below those the fatty acids. Now here's the thing. Fatty acids can show differences in length, how many carbons in there, and whether or not they're straight chain or, as you see in the figure over there, can have a double bond which gives them the bend. So, length, single or double bonds, are they saturated or unsaturated all the way through? Glycerol is always going to be about the same. Phospholipids is going to be the same. And then you see in the picture over there, at the top of the phospholipids, it says R. Well, what's the R's? R's can be pretty much anything. R's can be amino acids. R's can be sugars. R's could be a combination of the both. Turns out, saturated, unsaturated gives us one type of phospholipids. The R's that are just amino acids is another. R's that are sugar moieties. Moieties means different chain sizes, shapes of sugars, different sugar moieties. Give you another type, and then the mixture, all of the above. When we look at these phospholipids, sorry, that was my killer chihuahua. Ooh, that would be a great band name, Killer Chihuahuas. Anyways, the phospholipid bilayers. These are just a natural state of being for these phospholipids. Have to remember, the phospholipid head, the glycerol, the phosphate, whatever R group, are extremely water-philic, water-loving, whereas the tails, saturated or unsaturated, are extremely hydrophobic, water-fearing. So, what happens here? I give you a bunch of crystallized phospholipids, dried out. You dump them in water. What you're seeing, what will immediately happen, snap of your fingers, is this. They're going to form these spheres automatically. Notice here that the phospholipids right there are nice and protected from water, whereas these phospholipids, or these parts of the phospholipids, the heads are pointing out or are pointing in towards water.